Okay, it's a real pleasure to introduce today's speaker, Dave Begun, who I've known for a long time. I was a postdoc in Chip Quadro's lab at Cornell when Dave was a graduate student. And it was, for me, it was just an eye-opening experience to work alongside Dave. I learned a tremendous amount from him. So Dave made, as a graduate student, what I see as being one of the most important discoveries in empirical population genetics in the last 20, 30 years. I don't know when, I'm trying to remember when that paper was, showing that there's a correlation between recombination rate and levels of nucleotide variability across the genome. And he showed this in Drosophila, and it's now been shown in many other organisms. And the interpretation from that work was that that was due to the effects of selection at linked sites. And it spurred a whole bunch of research in trying to understand exactly what could drive it. Initially, it was interpreted as being due to positive selection, but then Brian Charlesworth suggested that it may also be due to something he called background selection, purifying selection, removing deleterious variants from populations. So that was a chapter in your thesis, I think.
Yep.
And I'm going to embarrass him now. And it was published in Nature, along with another chapter from his thesis, which was also published in Nature, and then there were a few other chapters.
It's been all downhill ever since.
So for the graduate students, that's something to aim for. And he spent much of the last 20 years working on the details of how selection shapes genomic patterns of variation, looking at environmental adaptation in Drosophila, and has also had a very interesting research program on the origin of new genes in Drosophila and other organisms. So he's had a very big impact in empirical population genetics. Now I can say that working next to Dave as a postdoc was both inspiring and demoralizing. Inspiring for the obvious reasons I just said, demoralizing because I knew we were going to go on the job market at the same time, and I didn't stand a chance next to him. Fortunately, he got a job very quickly, and so I didn't have to compete with him for long. His first position was at UT Austin. First he did a postdoc at Davis and then was at UT Austin, and then he's been on the faculty at Davis since when?
2000.
Since 2000, where he is in the population biology program. So today he's going to talk about environmental adaptation in Drosophila, and in particular about resistance to UV exposure. So welcome to the MVZ.
Thanks, Michael. Thanks to Michael, my old friend and one of my favorite colleagues. I miss harassing you, which it was fun. We should do that to each other more often. Yeah, and thanks to all of you for taking a little time out of your day to listen to some science. This is a new project that we're working on in my lab. And I think I was taught in grad school. I'm not sure. Maybe I was taught somewhere else that when you give a talk, you should try to talk about unpublished work, right? This is now a very quaint idea. So it does not apply. But so I'm apologizing in advance for telling you about this work, which came out last week in PLOS Genetics. For those of you that voraciously consume the population genetics literature, this is already old news. For the vast majority of you that haven't, you'll hopefully learn some new things about Drosophila biology. And before I forget, I want to thank the people in my lab that did most of the work. All postdocs. Nico Svetek was the first author on the PLOS Genetics paper and did a terrific job on many different aspects of this project. And then Julie and Lee both made really important contributions to the bioinformatics stuff that was done in this work. So before I dive into some evolutionary genetics, let me remind you in sort of the more abstract sense what population genetics is all about. And there are many ways to talk about this, but one can think about it as trying to understand the relative importance of these four processes or events in shaping genomic variation and in helping us understand the evolutionary process, the substitution of alleles over time. So it can be a very abstract and conceptual discipline. From the experimentalist's point of view, we like working with organisms because we like to understand how these abstract things that can be parameterized and theoretically studied integrate with biology. And we also hope to get some real life sort of ideas about how these theoretical ideas actually play out in the real world, right? Because real biology is always more complicated than theoretical models. So what does integration with biology mean? Well, there's a lot of ways of thinking about that, and the way I'm thinking about it for today is we really want to understand how the details of the biology intersect, in this case for today, particularly with how natural selection is acting on the organism. And this has become a very hot topic with the introduction of population genomics and the use of genome annotations to try to understand how environmental or other ecological variables, biotic or abiotic, might intersect with these processes of selection, combination, and so on. So I'm going to tell you about our attempt to try to get some biology into population genetics today in a new project. And we use Drosophila and melanogaster a lot in my lab. By the way, when Michael first asked me to talk here, it's like, you know I don't work on vertebrates, right? It's like, okay, yes, but we'll still allow you to come give a talk here. So I work on flies. You said I work mostly on flies. I work only on flies, just to be clear.
I thought you had an interest in snakes.
I used to be a boy herpetologist. But I'm going to tell you about melanogaster, one of my favorite species. It's got a lot of very nice attributes. It exists in large outcrossing populations, terrific genetics and genomics, as you know, a very large research community. All the aspects of animal biology that really you can find in a fly are studied by someone or multiple people in the fly community. And it's small and it's pretty inexpensive to keep. We can do some pretty big experiments, not on the scale of yeast or E. coli, but still, for an animal you can do some pretty big experiments using this organism. So it's got a lot of advantages. It's never really been a particularly good ecological genetics model. You can't go out in the field and study them very easily. You can't measure reproductive success of individuals in the field and so on. So it's not perfect. No organism is perfect for all biological questions, but it's quite good. And one of the reasons I'm so excited about the project I'm going to tell you about today is that I think it really is, it has a lot of potential to really start to use this fly as an ecological genetics model. Now, before I tell you about some data, I want to remind you about some of the salient properties of the species from a population genetics perspective. It's fairly polymorphic. It's got fairly low linkage disequilibrium. This means that if you do population genomics and try to identify plausible targets of natural selection in the genome, you can generally get good candidates down to the scale of genes, individual genes. So that's a nice thing. It's got an out-of-Africa history, much like humans. A lot of the differences in amino acid sequence between species seem to be the result of directional selection. Okay, so this is now approaching dogma in the field. There's debate about exactly what proportion of protein divergence between species results from directional selection, but it's a lot. It's a large proportion. That, by the way, is fairly remarkable, and no one really has a clue about understanding how or why so much adaptive protein evolution is actually occurring. I'm not going to talk about it today. Now, thinking about the segregating variation, most of the polymorphism in the species is influenced by selection at linked sites, either the removal of deleterious mutations or the spread of beneficial mutations. But one of the things we don't know much about is how much of the standing variation in the species is actually influenced by some kind of diversity-enhancing selection. So negative frequency-dependent selection or spatially varying selection. It's not really well known. It's a very hard problem, and so it's an interesting thing to try to think about. And population genomics is one of the ways that we hope to eventually try to get an answer to this question. Now, one of the things that is very clear in this species is that latitudinal declines are common. So they've been studied for many, many decades. Ever since the early 1970s, people have appreciated that this species shows latitudinal declines for several traits on multiple continents. So from that point of view, it seems like we can feel fairly secure in the notion that at least some of the polymorphism is maintained by some kind of spatially varying selection or local adaptation correlated with latitude. Here are just a subset of traits that have been documented as exhibiting strong declines on one or more continents. Many important life history traits, including body size, various temperature-related physiological phenotypes, diapause, and a whole bunch of other phenotypes, all broadly suggestive that spatially varying selection actually does play an important role in generating biological variation in this species. Okay, so from that point of view, it can be used as a model for trying to understand local adaptation. Chromosome inversions are also part of the story in this species. I'm not going to talk about those today. Now, from a gene-centric point of view, trying to think about understanding local adaptation from a gene-centric perspective rather than a phenotypic perspective, ever since molecular techniques were introduced into the study of this species, people have been interested in understanding whether there were declines. So when Allozymes showed up, people immediately tried to get some data bearing on the question of latitudinal variation for allozymes, and sure enough, a substantial proportion of them showed evidence of latitudinal declines, often on multiple continents. And then when RFLPs and Sanger sequencing came on the scene, again, people targeted some classic genes to try to understand whether or not they exhibited latitudinal declines, one of those classic genes being ADH, which actually had been documented as exhibiting an allozyme decline. And this obscure paper, which I published as one of the chapters from my dissertation, actually was a single locus study, which you could do back then and write a paper about, single locus study of a gene 6-PGD, which very much like ADH showed latitudinal declines on multiple continents and other interesting things. And I was interested in understanding whether or not the history of genetic variation at this gene was similar to that observed at ADH, because the patterns of allozyme variation, including the presence of latitudinal declines, was shared between these two genes. So I've been interested in latitudinal declines ever since grad school, and actually anybody who studies the population genetics of Drosophila melanogaster has to pay attention to latitudinal declines. It's a big part of the literature, and clearly is very important for their biology. Now, I kind of sort of left that area of population genetics, of spatial variation, after grad school. And there are many reasons for that, but when population genomic tools came on the scene, I immediately jumped back into this problem of spatial variation in Drosophila melanogaster. And it's very appealing to do population genomics, as many of you know, for several reasons. One of them is that you can get answers to some old questions, which I think that's happened in several systems. So, for example, it's possible, we don't really have a great answer in melanogaster yet, to try to estimate how much of the genome is perturbed by spatially varying selection, right? You could do that using whole genome approaches. Our current data sets aren't very powerful, but you can imagine being able to do that. That's pretty satisfying. That's a fact about nature that we might like to know the answer to. Then, I think the other big thing that's happening is that, and this is happening in many systems, humans and flies and lots of different plants and animals, is that by using population genomic approaches to try to identify portions of the genome that have been recently influenced by some kind of interesting natural selection, you're essentially letting the organism tell you, right, what phenotypes maybe you should be thinking about. Because when you integrate population genomics with genome annotation, you get this relatively unbiased view of how natural selection is shaping genomic variation, right? Whereas if you don't have that, if you're a fly biologist and you want to investigate this question, there are some phenotypes that fly biologists will tend to gravitate towards, right? So it's a very biased sample of phenotype space that you're looking at when you're just picking a phenotype because you think it's cool or interesting or whatever. The genome is giving you a less biased view of how natural selection has been perturbing the genetic variation. And this results in lots of interesting hypotheses, okay? Now, the problem is getting past the hypothesis generation stage to really deep experimental investigation of the forces maintaining genetic variation in natural populations. And it's a very hard problem. I'll tell you from my own experience that when I engage in this kind of activity, population genomics, trying to find evidence of local adaptation and so on, We find lots of interesting genes, that is, genes with very strong signals in the population genomics data for evidence of recent selection. And yet, when you look in the annotation, you're often faced with two different and equally severe problems. The gene, there's nothing known about it whatsoever. Nothing. Right? So it's one of your best population genetic signals in the whole genome, and there's nothing known about it. The other is, there's a gene, you've got some very interesting patterns of genetic variation, and you look at the annotation, which is often derived from knockouts, and the gene's very pleiotropic. So a huge number of different phenotypes are influenced, right, by the knockout. And when you look at the list of phenotypes, you're thinking, well, how am I supposed to think about a good hypothesis that I can test, right, as to how this variation is maintained by natural selection? It's a very challenging activity. Nevertheless, we jumped in with a couple of papers, 2008, 2010, 2014, and other labs have also attacked this problem of latitudinal differentiation. And one of the things that we observed in all of our data sets, including this early one, which was done by tiling array analysis, was that several DNA damage repair proteins seem to be among the outliers. That is, the genes that show the greatest latitudinal differentiation in our population genomic analysis. Now, one of those genes was MY41, which I'll tell you about, and it had the most differentiated, one of the most differentiated amino acid variants in the entire genome that we knew about. It's a key regulator of double-strand break repair. So we set off to try to see if we could get a little traction experimentally understanding what was going on with this gene, right? So this is the program. You do population genomics, you get an interesting biological hit, you think of some interesting experiments you can do to try to gain some insight into what's happening in nature. So one of the things we did, which we never published, is that because this gene is known to play a role in regulating the number of recombination events, crossovers occurring in the female germline, we were curious about whether or not it might be a modifier of recombination in nature. Now, why might that be under selection in clines? Well, the inversions, which are clinal, chromosome inversions, are gigantic modifiers of recombination, and they redistribute exchange events across their other chromosome arms. So we thought, well, maybe this is playing a role in interacting with the inversions. So we did a huge genetics experiment, very big, lots of flies counted, nothing. So no evidence of anything, never published that. And then the classic loss of function mutant, right, that fly geneticists work on in this gene, shows mutagen sensitivity. So you dump some nasty chemicals that are mutagens on the flies, and they do very badly. Right? So we did some experiments to ask whether this amino acid polymorphism in this protein affected mutagen sensitivity. And there was a little bit of evidence that that was true, but we never published that. And I kind of got a little bit disillusioned with this activity, because I'm thinking to myself, well, even if I get a good genotype-phenotype connection here, well, how's that going to help me understand what's happening in nature, right? I mean, mutagen sensitivity, it's, you could make up a story, but, it's not that satisfying. So we dropped it. Went on to some other things. Now, even back then, we were thinking about UV. But then we dropped that too. I'll tell you why. It wasn't a good idea to drop it, but we did. So UV is generally divided up into three different sort of wavelength categories. UVB gets through the atmosphere and strikes the Earth. UVC doesn't. UV8 gets through also. But UVB is of particular biological interest because that's the one that has the wavelengths that interact with double-stranded DNA. So it's a really big source of mutations. Okay? So you think, oh, latitudinal decline for DNA repair genes, UV, environmental, right, variable, worth thinking about. UVB induces two main types of lesions. Most of them are CPDs, cyclobutane pyrimidine dimers. These are like the thymine-thymine dimers that you probably heard about, right? The sort of most common kind of mutation induced by ultraviolet B, and then these 6,4 photoproducts. Both of these types of lesions are bulky, so they impede DNA replication and may lead to stalled replication forks. Stalled replication forks, if they collapse, result in double-strand breaks and cell lethality potentially. So it's a really big deal if the cell can't repair these UV-induced lesions, and so not surprisingly, there are multiple mechanisms that have evolved to repair this UV-mediated damage. One is the direct enzymatic reversal of these lesions. In flies, that's carried out by a protein called photolyase, and a lot of organisms, actually humans don't have photolyase, but a lot of organisms do. The predominant alternative mechanism of repair is called nucleotide excision repair. This is a process where the region near the lesion is unwound, and then the lesion is excised, and then DNA is chewed back and then resynthesized. So this is a repair process which actually involves DNA synthesis. Now, if you have these UV lesions which are not repaired by either direct enzymatic reversal or nucleotide excision repair, and you end up with a stalled replication fork, right, because the lesion is still there, the cells evolved another mechanism, a fail-safe, right, to fall back on, and that's known as translesion DNA synthesis. So at these stalled replication forks, there's the recruitment of this alternative mechanism of DNA replication, DNA polymerases and accessory proteins, that go to this location and then jump past the lesion and continue replication. Okay? Now this is great because that lets the cell escape essentially this mutation. But the thing is, after they bypass, they're very error prone compared to normal replicative polymerases. So there's a potential downside to the deployment of translesion polymerases in the cell. But nevertheless, the cells evolve multiple mechanisms for dealing with these UVB-induced lesions, and the reason is they're extraordinarily abundant. You can ignore this entire table that I took from a review article, but just this is in humans, peak hour, sunlight, DNA lesions from UV, 100,000 per day per cell. Okay? So, yeah, you are constantly experiencing enormous numbers of lesions as a result of UV exposure. Put on your sunscreen, right? Okay, so, drawing us back to the ecological issue, UV strikes the Earth, but importantly, it's latitudinally variable. So here's a heat map of UV that Nico put together. Or pulled up, I think he made this. It shows you that at equatorial latitudes, of course, you get very, very strong UV, and then you gradually diminish as you move away from the equator. So it's very strongly correlated with latitude. And here's some more quantitative data just to show you that these are yearly averages taken over many years from a bunch of ground locations near places where we ended up working on some fly populations, and it's just showing you there's a very strong correlation between latitude and UV. And then to sort of give you a feel for so this figure basically contrasts a population at 5 degrees latitude and one at 45. We wanted to get this one in because we actually, as I'll tell you in a moment, worked on a population roughly at 5 degrees latitude. So threefold different amount of UV near the equator versus a high, threefold different. Now, can you think of any other abiotic variables that vary by threefold with latitude? I can't. It's pretty striking ecological gradient. So that sounds good. There's a latitudinal decline for DNA repair proteins. There is UV that varies latitudinally. You know, it sounds like a good story, but the cuticle protects the fly from DNA damage. So that's what we were thinking. Well, the adult is protected. Very little UV gets through. The larva has a cuticle. It's protected. The larva burrows in rotten fruit. It's not getting exposed to much UV. So we thought, well, you know, it's just not very relevant for the animals, even though there's a latitudinal decline. So we stopped thinking about it. Then when Nico came to the lab, we started thinking about the early embryo. And that is a totally different story. First of all, it has no cuticle. It's just got a very thin layer of cells, and UVB energy penetrates those cells and gets into the embryo. It's got a very fast rate of genome replication. So these fly nuclei during early development in particular are replicating as fast as animal DNA can replicate. Right? They are really right up against the limit of what DNA replication can do in an animal. Okay. That leads to what's known as endogenous replication stress. So these early embryos are undergoing very rapid nuclear divisions, and if you add a UV damage on top of that, you could see how it could be a big deal to the viability of the embryo. My slides look a little funny, but that's okay. The other thing is that at a particular stage in development, so flies do this weird thing in development where their nuclei keep replicating, but there's no cellularization that occurs. So at one point during early development, all of these nuclei migrate to the periphery of the embryo, and then cellularization occurs. Okay? But when they're at the periphery of the embryo, you can imagine that they're right up against where the UV is going to get through the chorion, right? So you can see that there really may be some UV sensitivity, and of course embryos can't move, right? They can't choose their environment, so they're exposed to whatever abiotic or biotic gradients are present in their environment. So we set out to see if there was any geographic variation in embryo UV sensitivity. We were curious. Okay? So we sampled a series of populations from Panama to Maine. These are fresh samples that we had established in the laboratory. And a fairly large number of lines and a fairly large number of embryos. In this experiment, Nico et al. Assayed hatch rates for over 50,000 embryos. So that's what I mean by you can do pretty big experiments in this organism. And the experiment that we did first was simply to compare the hatch rates of the embryos in a control set of experiments and a set of experiments where we exposed the embryos to UV. So here's how we do this experiment. We blow up our stocks, we expand them so we have a lot of females, we collect embryos on plates, and then we put them on these little Baskin-Robbins spoons. All fly people use these things. They're scattered all across the hall in the building I'm working. There's a little bit of food in the spoon part. Right here, these little white things are the embryos. So you can move them around with a dissecting needle. It's pretty easy to do. So we would take from each strain, each of our 111 strains, we would collect a bunch of early embryos. They were all aged 1 to 3 hours. Then some of them would be shunted into our control treatment. Some would be put into our homemade UV exposure apparatus. It's a box. It's a box with a UV lamp and a little fan to maintain the temperature at ambient level. And we expose the embryos. And then all of the spoons, control and experimental, got put into food vials, and then later on, the hatch rates were actually just estimated by direct observation. Okay, so that's how we measured. That's how we measured things. And for each line, for a total of 6 populations, we estimated embryo UV sensitivity, and that's simply the difference between the control and the UV exposed hatch rates for these embryos. And there's a beautiful latitudinal line here, very significant, even though there's only 6 data points. Okay, and latitude explains a lot of the variance. The slope of this cline is comparable to that observed for many other phenotypes in this species. Note that the low latitude population is the most resistant to UV at the early embryo stage. So that's kind of what you would predict if the story that we're telling ourselves, if there's something to it. This is the same data set, just with the line data represented, so you don't see, there's not as much variance, of course, explained by latitude when you look at your data this way, but it's very, very highly significant because there's so many lines. So no matter how you look at the data, there's very strong support for latitudinal decline for early embryo UVB resistance. Okay, so what I'm showing you here is, instead of showing you the deviation of the control from the experimental hatch rate, I'm just plotting the results separately for the 2 experiments, control experiment and UV exposed. The blue is UV exposed, and this green is the control hatch rates. So the weird thing about this, which I was actually a bit surprised about, was that both experiments showed evidence of latitudinal declines. This is, I think, the best evidence that we have of just a latitudinal decline for just regular embryo hatch rates with no experimental treatments whatsoever. So the way I think about this is that the low latitude Panama population is the most resistant to UV, but in the absence of UV, it has the lowest hatch rates. So you can kind of start thinking about this as at least generally consistent with the idea of trade-off, although by no means, of course, does this demonstrate that there's a trade-off. But perhaps the adaptations, if there are adaptations associated with embryo UVB resistance at low latitude populations, that there's a cost to those adaptations and that it reduces the hatch rate of embryos when it's like a classic resistance phenotype, right? In the absence of the agent of selection, the fitness of those alleles might be reduced. Okay, so that's all great. We're starting to feel really good at this point because we told ourselves a story and it looks like there may be something to this story. But there was one thing that was a little bit strange, which is that flies are very strongly positively phototactic, and that includes gravitating towards UV. So they go towards UV. They go towards visible light, and they go towards UV as well. And we thought, well, that's weird. We wouldn't necessarily predict that, and yet it's one of these classic results, decades-old results from the literature. Then we were sitting around the lab, and we said to each other, well, you know, if we're right about this whole thing about selection on the embryo from UV, then female flies that are getting ready to oviposit, when they're looking for a place to lay their egg, they should actually avoid UV environments, right? Because UV is damaging to their embryos during early development. So we said, we would predict that when females are choosing a place to lay their eggs, they should avoid UV. And this kind of blew me away because a couple of months after we had this conversation, this paper came out from a neurogenetics lab at Duke. We've actually tried to establish since then a collaboration with the PI of this lab. But they showed in this experiment that, in fact, flies are positively phototactic, just like everybody says they are. But when the females are physiologically competent to oviposit, the preference flips 180 degrees. When they are getting ready to lay an egg, they avoid UV. And that is completely consistent with our ecological hypothesis about UV and its interaction with early embryogenesis in nature. So that was a fun moment when that paper came out because we had predicted actually that should exist. All right, so if you follow me so far, we have latitudinal declines for repair proteins. Then we actually showed that there's an early embryo UVB resistance phenotype that varies geographically. So one thing you would predict from our model is that there should be geographic differences in DNA repair. That is, there are multiple mechanisms for why we might have seen our UV embryo resistance climb. But we are specifically proposing that there should be geographic variation in DNA repair. So we set out to get some insight into this, and we actually haven't demonstrated yet that there's geographic variation in repair. But we did an experiment which very, very strongly suggests that there is. And that was a genetics experiment. So we haven't measured DNA damage directly in the embryos and the repair of that damage, but we have done an experiment which I think is pretty cute and supports our hypothesis. Now, before I tell you about this experiment, you have to appreciate one thing about what's going on during early stages of embryogenesis. It's strictly maternally determined. So all of those events during early embryogenesis are essentially, you can think about them as a phenotype that is the result of the mother, right? What she puts into the egg. So it's the maternal genotype and the maternal effect during early embryogenesis. So the other thing you need to know is that in flies and other animals, I don't know all other animals, but other animals, the oocyte actually can repair damaged sperm DNA, right? So after the sperm enters the egg, damaged DNA in that sperm can actually be repaired in the oocyte. So we took advantage of this to ask the question, are the oocytes of females that are associated with high embryo UVB resistance, are they exhibiting more DNA repair? Okay, so that's the experiment that we're going to do, and here's how the experiment works. Okay, so we take a strain of flies carrying a balancer that we can follow through a cross. We take males from that strain. This is an X chromosome that we're following through the cross. And we expose these males to mutagen, classic mutagen used in mutagenesis experiments. So there's lots of damage being introduced into the sperm, right? Into the genomes carried by these sperm. All right, so we imagine a female from one of our isofemale lines, and we mate this mutagenized male to this female. The sperm enters the egg, and some repair of damaged sperm DNA is going to occur in the egg. Now remember, this damage should lead to reduced viability associated with this mutagenized chromosome, right? Chromosomes that are mutagenized to a greater degree should be associated with lower viability in the resulting zygotes. Okay, this sperm enters the egg, some repair is going to occur. Then we recover that mutagenized X chromosome in this female, okay? And we mate her to her brother, and then we look at the F2s. So what we're doing in this experiment is basically counting the proportion of these balance of chromosomes that were mutagenized in the grandfather, right? And some of those mutations were repaired in the egg of the grandmother. And what we're predicting is that if we take a whole bunch of females that are exhibiting embryo UVB resistance, and we compare them to a whole bunch of females that have UV-sensitive embryos, that we should recover these mutagenized X chromosomes at a different rate. And to be specific, for lines associated with very strong UVB resistance at the embryos, we should recover more of these mutagenized chromosomes because more of the damage was repaired in the oocyte, right, of the grandmother. So we took from our 111 strains, our 14 most resistant and 13 most sensitive, so the tails of the phenotypic distribution, and we did this experiment. And here's what we saw. So we scored over 60,000 F2s, and we saw a very significant difference between the resistant strains and the sensitive strains. The resistant strains, we recovered about 7% more of these DNA damaged balance of chromosomes. So this is the best evidence we have to this point that there is a correlation between strains that have very resistant embryos, that is UVB resistant embryos, and DNA repair. There seems to be pretty strong support for that. We still want to do some more explicitly geographic and molecular genomic approaches to try to directly quantify damage at the DNA level, but we haven't done those yet. But this supports our hypothesis that there's geographic differences in actual efficacy of DNA repair in these Drosophila populations. And tracing all the way back to our population genomics data, it's consistent with the differentiation we observed in the DNA repair proteins. Okay, so following along in this line of reasoning, there are two ways you could imagine low latitude flies have adapted to high UV environments. One is that they might have protein variants with differential enzymatic activity, for example, greater efficiency or rates of repair, so amino acid variation. But another could be simply dosage effect, right? Low latitude females may simply be putting more DNA repair transcripts, and then, of course, you get more protein, right? You think. They might be allocating more DNA repair transcripts into their embryos, eggs, during development. So we wanted to see if there was a sort of expression difference in the early embryo between high and low latitude females. So we used our Panama population and our Rhode Island population, and we took the same aged embryos that we used in our UV embryo experiments. We made pools from several lines from each population, and we had 3 biological replicates for each of these pools, and then we did a sort of standard, these days, standard RNA-seq analysis on the 6 libraries. Okay, so we generated a bunch of data, and then we're thinking sort of what we want to do with it exactly. Now, obviously, you have whole transcriptome data, but to be honest with you, at this point, we kind of were past the point of looking in the whole transcriptome for differences between the high and low latitude populations. We weren't interested in that. I mean, yes, I'm sure there are many interesting things in the data, but we are really now starting to focus in on our phenotype and the candidate genes that might be responsible for this phenotype. So we're thinking about DNA repair proteins now. So we wanted to generate a candidate gene list. So the melanogaster genome is very nicely annotated. We picked several geoterms that we thought were reasonably associated with the phenotype that we're interested in, this DNA repair during early embryogenesis. The DNA repair literature is massive, by the way. It's absolutely huge because of the role of these genes in cancer biology. And so depending on how you pick your gene ontology terms, you can get a bigger or smaller list of quote-unquote DNA repair proteins, but we were pretty picky about the geoterms we used. And then, on top of that, we did manual curation. So I don't know if any of you do a sort of dumpster diving into the gene ontology sort of stuff. Sometimes it's a little frightening how a gene ontology term gets attached to a gene because the evidence on which that's based is so flimsy or tangential that you can't understand how that gene ontology term got attached to that gene. So I'm not blaming the gene ontology people. They're great people. To quote Trump, right? Great people. Great people. Great people. But you've got to. Well, we thought was worth manually curating our list, and when we did that and looked at the papers a little bit more deeply, we winnowed that list down to a total of 211 genes. We felt pretty good about it. I'm not saying we didn't miss some that were relevant, but this is a pretty manageable collection of candidate genes. Actually, I talk about this is like the Goldilocks trait for me now because it's just right. It's not some bizarre yet highly interesting trait, don't take offense, where the genetics is extraordinarily simple, right? Like there's a pigmentation mutation. It's like, love that stuff, it's great, but probably most adaptation isn't like that. And yet it's not so highly polygenic, like 1,000 genes, that it's almost hopeless to imagine how a normal person could go about trying to understand it from an experimental point of view. 211? I can handle that. That's pretty good. I can do experiments. I can have really well-focused hypotheses about those 211 genes. So I wasn't shooting for a number like that, but I was pretty happy when we got to this number because I think I can work with that. Okay, now, getting back to the transcriptome differences between Panama and Rhode Island embryos. These are early embryos, 1 to 3 hours old. So in that data set, we had about 8,500 genes that were expressed, and we had about 856 that were differentially expressed. Now, of our 200 expressed candidate genes, so 211 candidate genes altogether, 200 were expressed, 21 were differentially expressed. So Nico and I were talking about this, like, well, there's no enrichment for differentially expressed DNA repair genes. It's like, I don't care about geo-enrichments. I'm past that. It's like, we are focused on our phenotype. We have other phenotypic data that support the importance of this phenotype and the geographic variation in this phenotype. Let's use the data in some other way. Let's ask about directionality. And that's what we did. So here is the directionality, I'll explain to you that in a minute, of all the differentially expressed genes in the experiment, 856. So 489 of them showed greater expression in Rhode Island, and 367 showed greater expression in Panama. So it's not one-to-one, and I can sort of, there are possible technical biases that might be the cause of that, or maybe it's a real biological thing, I don't know. But the crucial thing is if you compare all differentially expressed genes and their directionality to the directionality of our candidate genes, it's very, very heterogeneous. So of the 21 differentially expressed genes in the early embryo, right, Panama versus Rhode Island, 20 of them showed higher expression in Panama. Very, very highly skewed relative to overall transcriptome differences. Very significantly heterogeneous, one of those minuscule p-values, if you assume that these are independent, right, mechanistically from a gene regulatory point of view. Now, we don't know if that's the case yet, but I think there's a decent chance that it will be the case because there's a lot of evidence of cis-acting variation in flies, so for many different genes, so probably for these too. Okay, so of those differentially expressed candidate genes, several of them are associated with nearby non-coding SNPs that are also very geographically differentiated. So, those are plausible candidate SNPs for driving the geographic differences in gene expression. And we're currently doing some genetic experiments to actually get a handle on how much of the geographic differences in expression are the result of nearby cis-acting variation versus some sort of unlinked transactive variation. So we're doing those experiments right now. Okay, so so far we have a journey that we started on from population genomics data, suggesting DNA repair differences between high and low latitude populations. We then showed that there was a latitudinal decline for early embryo UVB resistance, and UVB is a main source of DNA lesions in natural populations. We then showed that there was a very nice correlation between fly lines that had highly resistant embryos and those that could repair damaged sperm DNA, supporting the idea that there's actually underlying this latitudinal decline differences in DNA repair, which is consistent with our original observation of latitudinal declines in DNA repair proteins. And then we showed that there were transcriptome differences between high and low latitude populations for our DNA repair genes that are very strongly suggestive that low latitude females, one of the adaptations is that they allocate proportionally more DNA repair transcripts to their eggs, right, when they're making them. So that's what we know so far. Now returning back to the population genomics, our early population genomics data, which got this whole research line off the ground, was a comparison of Maine and Florida, or sometimes Queensland and Tasmania. But you may have noticed that we had a lot of experimental work from our Panama population, but no population genomics data. So we generated some. We generated about 80x worth of data from a pretty good sample, 100 independently isolated chromosomes from a natural population down there. And we did sort of the really obvious stuff that you would do if you were engaged in this kind of activity. We estimated what I call genic SNP differentiation between Panama and Maine. That is SNPs in or near genes. We used FST for that. This is definitely not state-of-the-art kind of stuff, but probably good enough for what we're wanting to do right now. And then, again, we're not really, I am interested in the whole genome differences between Panama and the high temperate populations, but we haven't actually even done a whole lot of analysis on that yet. We're focused again on our 211 candidate genes because we want to gain some more insight into what's going on there. So what we observed in terms of protein variation in those 211 genes is that roughly 60 of them had at least one very strongly differentiated protein variant. So, is that an enrichment relative to genomic background? I don't care. And I encourage you not to care about enrichments like this, because remember, we're really now investigating in a very focused way a particular phenotype and a collection of genes that are likely associated with that phenotype. So 60 candidate genes with a very strongly differentiated non-synonymous mutation, or more than one. So, I'm not going to show you a gene list, but there's a ton of interesting proteins in there, and a lot is known about them because of their role in cancer biology. So this project is very rich in potential. And what I do want to do, instead of showing you a gene list, is, this is one of those slides. You can just space out and listen to me if you want. Don't try to read the gene names. That's totally pointless. So what I've done here, and we're going to ignore these two bubbles here, okay? This is a figure from our PLOS Gen paper. So what I've done is I've organized the various processes in DNA repair that are probably relevant to the early embryo in nature for this UV phenotype. So the lesions need to be recognized. There are a couple of genes that show differentiation. Nucleotide excision repair, which I told you about, stalled replication, fork stabilization, and then translesion synthesis. So the way this figure is organized, the genes in black have a non-synonymous outlier SNP, okay? One or more. The genes in blue show expression differences between high and low latitude populations, and the genes in green show both expression and protein differentiation. So really the take home from this kind of figure is that, first of all, multiple components of different parts of the whole process of DNA repair seem to be influenced by spatially varying selection. So that's one conclusion. Another conclusion is there are many genes in which there seem to be both non-synonymous differences and expression differences. So maybe there's something going on here, just like that classic gene ADH, where natural selection is acting on total activity, and that can be influenced by either the amount of transcript or protein produced or by tweaking the protein sequence to change the activity. So anyway, there's a lot of experimental opportunities in here and a lot of interesting things to think about. And that's really the big take home, that multiple big and important components of DNA repair seem to be influenced by selection, and both protein and expression variation seem to be targets of selection. At least we think so. Okay, so what kinds of issues can we address? Lots of interesting ones. So we can start thinking about, in this system, the relative importance of protein versus gene expression variation. We can start thinking about effect size, that is the effect on our phenotype versus population genetic parameters, right? So one of the great things about population genetics data is it should be telling you something about fitness variation in nature. We cannot measure that in flies. We can't measure fitness variation in nature. It's really hard to do. Although I can put embryos, right, out in sunlight and test various hypotheses, which we haven't done yet. So I think actually we can do some experiments in nature. But we can measure the effect size on our phenotype. And if these two things don't go together very well, it's telling you something, right? It's telling you maybe that there's some pleiotropic effects that you don't know about. So that the effect on your phenotype is only part of the story, and there are pleiotropic effects on other phenotypes that alter the selection coefficients. So pleiotropy is something we can really attack, and we can also attack how the position of a gene in a pathway or its physical interactions may influence what kinds of alleles may be under selection in this kind of system. And really intriguingly, although I'm not going to go into it, lots of these candidate genes and others not shown in this figure interact physically. They're known to interact physically with other candidate genes. So there's a lot of interesting experiments to do about protein-protein interactions in this system. In this system, we can ask about the genetic properties of the alleles, like their dominance and their interactions with other genes, and then we maybe can start thinking about testing some models of polygenic spatial adaptation that have been published recently. So I'm pretty bullish about that. Now, getting back to the main question, why we started on this kind of science, what's responsible for the maintenance of variation? I don't know. But I think I can try to answer that. So, following some of the reasoning I presented earlier, there may be a cost of DNA repair, right? So low latitude genotypes that allocate all of this extra DNA repair capacity, in the absence of all this exogenous DNA damage, those might be disfavored. Those high repair genotypes might be disfavored by selection. Why? One possibility is the metabolic cost of allocating all those extra transcripts to the egg. To me, that seems like it's unlikely to be a big deal, but it certainly could contribute. More interesting to think about is the phenomenon known as gratuitous repair. Gratuitous repair occurs when DNA repair complexes are basically ectopically acting on undamaged DNA and essentially causing DNA damage and problems for the cell. So you can imagine if you had all of this extra repair capacity, right, in your oocyte, but you weren't experiencing a lot of damage, it might be spuriously allocated to undamaged DNA, leading to problems with nuclear division and embryo viability. And then another thing that we're thinking about is these translesion syntheses, these translesion DNA polymerases, which are big players in our candidate genes and our expression data. They do have high mutation rates, so they're very error prone. So maybe during this bypass process, there's a lot of mutations being introduced to the embryo during early development by all of these error prone polymerases. And in a fly, you can't ignore genotype by temperature interactions as part of the story. So, I think there's a pretty solid case at this point that UV is a very relevant agent of selection in natural populations on the early embryo, and it raises all kinds of interesting questions about other traits, besides embryo traits. One is where are the females laying their eggs, right? That might be under selection. Females may be able to assess their UV environment and allocate repair transcripts to their eggs specifically. Genome size is something that we're interested in looking at, because you might imagine at low latitudes, big genomes should be disfavored, right? Because there's going to be more damage per nucleus in the big genome. So that could be an interesting thing to look at. And then a lot of our candidate genes are expressed in females, in the ovary, and a lot of them play a role in meiotic recombination and DNA repair, obviously. So perhaps selection on the embryo is influencing differences in meiotic recombination or mutation. And there's other questions relating to altitude and different continents, which we're interested in, and then there are potentially some interesting comparative aspects. And I see I've used up all my time, so I'll stop there, and I'd be happy to take any questions, even though it's pretty late.